Welcome students. My next lecture on embryonic development. We already study in within 5 minutes, that is how the zygote is formed during fertilization process. After fertilization, the next process, that is embryonic development. What is embryo? The formation of or the development of the zygote up to the embryo in pregnant woman. Okay, 2 to 8 weeks child or baby is known as embryo. Before this it is zygote. We are still in reproduction, lower and higher animals, part 13, the embryonic development. Embryonic development, see here, all these are the development stages of the zygote. Zygote is a single cell but a diploid cell. Okay, after fertilization, about 12 to 15 hours, what happens? Two cells are formed. Zygote is divided into two cells, then two cells divided into four cells. After 24 to 30 hours, then 8 cell stage, then Morula stage, early Blastocyst stage, and late Blastocyst. I show here the diagram for your convenient study. Okay, 2 cell, 4 cell, 8 cell, Morula, early Blastocyst, late Blastocyst. All these happens only because of the mitotic division of the zygote. Zygote undergoes mitotic division. That process is known as the cleavage. See here, holoblastic cleavage. What is the meaning of holoblastic cleavage? First, we must know what is cleavage. It is the process of mitotic division of the zygote into multicellular blastula. What is blastula? Is a ball of cells. Multicellular ball, that is the blastula. Mitotic division of the zygote into multicellular blastula. And holoblastic, okay, in animal there are two types of cleavage. Incomplete and complete. Okay, incomplete or meroblastic cleavage, where the zygote is not completely divided. For example, birds and reptiles. In case of eggs of birds and reptiles, you know about the hen's egg. Okay, what are the structures present inside the hen's egg? You know the bulk, yellow color bulk, that is the yolk. Okay, on this yolk there is a whitish part, that is the process of development of the zygote, which do not completely divided due to more amount of the yolk. Meroblastic incomplete cleavage is due to because of more amount of the yolk. That such type of egg is known as megalocithal or macrolocithal, while the human egg is microlocithal. Already I told in the first lecture. Okay, female egg is, in case of human being, macrolocithal, very small amount of yolk. So here is the complete division of the zygote takes place. Zygote is completely divided. So the holoblastic word, holo means complete, and such cleavage is known as holoblastic cleavage. Okay, while in other cases there is an incomplete, that is known as the meroblastic cleavage. Holoblastic cleavage. See here diagram, which cleavages are take place. Zygote, a longitudinal cleavage or meridional cleavage. Another name for the longitudinal, that is meridional, first cleavage. Then second cleavage is also longitudinal, but it is right angle to the first. This one is a longitudinal, another is also longitudinal, but it is right angle to the first one. And the third cleavage is the horizontal. Cleavage. All these cleavages converts the zygote, divides the zygote into multicellular ball-like structure, that is blastula, and the process of formation of blastula is known as blastulation. See here how the blastula is formed. First cleavage, 12 to 13 hours. Longitudinal cleavage divides the zygote into two cells, that is known as blastomeres. The dividing cells are known as blastomeres. After division, the cells are known as blastomeres. There is no increase in the size of the zygote. Okay, only division takes place, no doubt, but successively the cells decreases in size, but the metabolic rate is increases. Okay, see here blastomeres, two blastomeres. two-cell stage. First cleavage divides zygote into blastomere, two-cell stage. Next process of blastulation. After two-cell stage, second longitudinal cleavage which is right angle to the first one, and this two-cell stage converted into four blastomeres, four-cell stage. See here, four-cell stage. Four-cell stage. First cleavage, second cleavage is also longitudinal, but it is right angles to the first one. Now four-cell stage converted into eight-cell stage because of the third cleavage, horizontal cleavage, which divides four cell into eight-cell stage. Now the number increases. Now they further undergo the mitotic divisions and form 16 to 13 celled stage, and these stage looks like a mulberry fruit. So the name here, Morula stage, looks like a mulberry fruit, mulberry plant. You already studied in sericulture or maybe heard in sericulture silkworm rearing. Okay, this silkworm feeds on the leaves of the mulberry plants. Okay, and these plants having a fruits which that is the mulberry, and this stage looks like a mulberry. So here words morula, which is the 16 to 13 celled stage. This morula further undergoes to form blastula, that is blastulation process. See here, morula stage, 16 cell stage. Out of these 16 cells, some cells are small and some cells are larger. The larger cells are in the center, while the periphery are the smaller cells. These smaller cells at this stage, all these stages are found in the female reproductive tract. From ampulla, these cleavages taking place in the oviduct region, first day, second day, third day, fourth day. When fourth day, at this stage, morula stage, come into the isthmus region. At this isthmus region, what happens? Zona pellucida is not breaking down. It is intact. Okay, it is intact. It is most important for this stage. If the zona pellucida breaks, then the implantation, that is the attachment of the stage, take place anywhere, or maybe abortion takes place in that woman. Okay, so zona pellucida must be intact during this stage, morula stage. This is the zona pellucida, and inside what is 16 cells are present. Of these 16 cells, periphery cells are smaller and the innermost cells are larger. This morula takes the glycogen rich uterine milk. After taking this nutrition, the space created and the cells which are smaller, they goes to the periphery and they form the outer layer of cells, and which are the larger cells, they are goes towards the one pole of the cell. See here, the cells which are smaller, they form the outer clear cells, thin cells, and these cells form outer layer of cells, and see here, that is known as early blastocyst, and the innermost cells which are larger, that forms the inner mass of cells. Now early blastocyst, see here, this early morula converted into early blastocyst. What happens? Inner cell mass and outer cell mass layers are formed. Inner cell mass, the point at which they get attached to this outer mass of cells, that is embryonal pole, and the opposite side, that is abembryonal pole. The name indicates embryonal pole, that is in that side this inner mass of cells form in future embryo, okay, baby, and outer layer of cells, they form the protective membranes of the embryo. Early blastocyst. Late blastocyst. From morula up to the late blastocyst formation is known as blastulation. Blastulation is the process of formation of the blastula or blastocyst, is known as the blastulation. What are the structures found in the blastula? Late blastocyst. See here, no doubt inner cell mass. A space get created due to taking of the nutritive fluid. This pressure of the fluid, due to pressure of the fluid, inner cell mass gets attached on one pole, and that pole is known as embryonal end, and the other pole which do not contain this inner mass of cell, that is known as abembryonal pole. See here, abembryonal end. Embryonal end. The end indicates that there is the attachment of the embryo, inner cell mass and the outer cell mass. Okay, outer cell mass is known as trophoblast, and the cavity which is created is known as blastocyst cavity. In this cavity, the uterine milk get absorbed. Late blastocyst. See here. After blastulation, that is gastrulation, formation of gastrula. Okay, which is the most important step. What is gastrula? What is gastrulation? Formation of gastrula, no doubt. In this step, what happens? The cells form the three germinal layers. We know the germinal layers in the animal kingdom. Trigeminal, that is the three germ layers, outer ectoderm, middle mesoderm and inner endoderm. When all these three germ layers are formed, and from these germ layers organs are formed. Okay, when these three germ layers are formed, that stage is known as gastrula. From blastula, gastrula is formed, and the process is known as gastrulation. How it takes place? See here in the diagram. Outer trophoblast layer, that is outer mass of cells in blastocyst. Okay, when gastrula is formed on 8th day, okay, before 8th day, on 7th day after fertilization, okay, these blastula get embedded in the uterus. After 4th day, after fertilization on 4th day, morula stage, which is come up to the isthmus part of the oviduct, but this morula reaches in on 6th and 7th day, and that day they get deeply embedded, that is blastula get deeply embedded in the uterine wall. Uterus having three walls: outer perimetrium, middle myometrium, that is muscle, smooth muscle, inner endometrium, it is richly vascular. We already studied about the endometrium in case of female reproductive system as well as in menstrual cycle. Endometrium contains endometrial glands which secrete the uterine milk, which is the nourishing fluid for the embryo. Okay, see here, they get embedded in the endometrium. At which place actually they get attached? Okay, these blastula or gastrula, where they get attached? They get attached the posterior superior wall of the uterus, fundus region. Uterus having three parts: upper dome shaped fundus, middle body part, and the last part which opens into the vagina, that is the cervix part. They get attached into the posterior superior wall of the fundus region. On 7th day, they get completely embedded in the uterine wall. How it is possible? Because of the trophoblast layer, outer mass of cells which are smaller in size as compared to inner mass of cells. This is known as trophoblast layer. Tropho means food, blast means layer. Tropho means food and blast means layer. Food layer, because this layer provides the food. This layer absorb the fluid, uterine milk, from the endometrium of the pregnant mother. Okay, when they get completely embedded, deeply embedded in the endometrium, this trophoblast divide into two layers: outer syncytiotrophoblast and inner cytotrophoblast. What is the meaning of syncytiotrophoblast? That is, these trophoblasts having the only plasma, okay, and cytoplasm and nuclei, not any cell boundary, cell membrane. Without cell membrane, these trophoblast layer, these trophoblast cells do not have cell membrane, only layer of the cytoplasm, and contains many nuclei, multinucleate trophoblast. That is syncytiotrophoblast, means together, okay, cytoplasm together with the nucleus without cell membrane. And these syncytiotrophoblast get embedded deeply inside the uterine wall. And one more characteristic of this trophoblast, that is, it is phagocytic layer. Okay, it is phagocytic sticky layer. That's why zona pellucida do not get disturbed in morula stage. Okay, when they get deeply embedded, at that time zona pellucida breaks. Okay, and this sticky trophoblast, due to sticky nature, they get embedded in the uterine wall. Second layer of the trophoblast, cytotrophoblast. These trophoblast cells having the cell boundary, they having the cell membrane, cytoplasm and nucleus. Okay, outer seed cytotrophoblast, only cytoplasm with many nuclei. They are sticky in nature, and with the help of seed cytoplast, they get stick to the attached to the implantation means attachment, implanted in the fundus part of the uterus. And cytotrophoblast having the cell boundaries. Due to uterine fluid pressure, these cells become flat. Okay, they becomes flat due to pressure of the fluid. That is cytotrophoblast, flattened cells. What about the inside? A cavity is created. We already studied in the previous slide, that is blastocyst cavity. Blastula contain blastocyst cavity. Now, how gastrula is formed? Some cells of the inner mass of cells, okay, at free end, they form the endoderm layer. At the embryonal pole, here the inner cell mass, the free end of the inner cell mass form the primitive endoderm, that is known as hypoblast. This endoderm grows towards the lower side and forms a sac, that is yolk sac. Yolk is the nutritive part. Okay, yolk sac. We already studied in the development of the ovary, yolk sac. You should see that slide. Then ectoderm. How ectoderm is formed? The remaining inner mass of cells form the epiblast or the ectoderm. Here, ectoderm is formed. First, endoderm, hypoblast, which are present at the free end of the inner cell mass, and the inner cell mass layers, cells which are attached to the trophoblast, that cell forms the ectoderm, epiblast. Both these cells form the bilaminar layer. Okay, bilaminar layer. Okay, and this bilaminar further converted to trilaminar, that is third layer is formed mesoderm. Now they undergoes the morphogenetic movement. One more characteristic of gastrulation, that is they shows the morphogenetic movement. The cell shows movement, okay, and forms the layers, outer ectoderm, inner endoderm, and middle mesoderm. From this bilaminar, the cells divide, redivide, shows the movement, and form the third layer, that is mesoderm. These ectoderm layers get divided and encloses a cavity, amniotic cavity. The upper surface of the amniotic cavity form a amnion layer. It is the extra embryonic membrane of the embryo. Okay, amniotic cavity, amniogenic cells or the amnion layer, and here is the ectoderm layer. And endoderm encloses the yolk sac, and in between two mesoderm layers, mesoderm forms two layers, inner mesoderm layers and outer mesoderm layers. Between these two layers, what is present is cavity is present, which is filled with the coelomic fluid, that is coelom. We studied about the coelom in the animal kingdom in 11th standard. Okay, acoelomate, pseudocoelomate, coelomate animals, hemocoelomate, coelomate animals, those animals which contains the coelom. True body cavity. What is the meaning of true body cavity? Okay, that is 3 germ layers, no doubt. In when a cavity is present in between two mesoderm layers, outer ectoderm, outer mesoderm, then a cavity filled with the coelomic fluid, true body cavity, inner mesoderm and endoderm. Such arrangement, that is coelomate animals, coelom. And further, these since cytotrophoblast further in future form the chorion layer, which helps to deeply embedded into the endometrium wall of the pregnant mother. This is all about the formation of 3 germinal layers in case of gastrula. Okay, formation of gastrula, that is gastrulation. See here, how it happens in the female reproductive tract. In ampulla, a site of fertilization, sperm and ovum get fused and forms the zygote. Zygote undergoes cleavage, a mitotic division. First cleavage form the 2 blastomeres. First day, second day, 12 to 30 hours, 30 hours, after 30 hours, second day, after 4th day. 16 to 13 celled stage, morula. It comes into the uterus. The structure that is early blastocyst, late blastocyst. This is the fundus region of the uterus where the implantation takes place. Implantation takes place, that is attachment of the blastocyst to the uterine wall, endometrial wall. Okay, and the uterus contains three parts: upper dome shaped fundus, middle body, and the lowermost cervix, which opens through outside to the vagina. And uterus contains three layers: outer perimetrium, middle myometrium, inner endometrium. Okay, they get deeply embedded in the endometrial wall, that is known as implantation, which takes place on the 7th day after fertilization. It takes place when implantation takes place. After fertilization, on 7th day implantation takes place. Late blastocyst get attached to the, it becomes 1.5 to 3 millimeter in size. Blastula get converted into gastrula. Here the frontal section of the endometrium of the uterus showing the blastocyst about 8 days after fertilization. Actual gastrulation process starts after fertilization on 8th day. Okay, 10 days after it is completed. See here, this is the endometrial wall, fundus region. Endometrial gland is present, innermost layer, endometrium of the uterus. Here syncytotrophoblast, which show only cytoplasm boundaries with many nuclei. Syncytotrophoblast, which is sticky in nature. Due to sticky nature, they get stick to the endometrial wall. And inner cytotrophoblast, which is the flat cells, which is made up of flat cells. Then amnion, amniotic cavity. Amnion layer is in contact with the cytotrophoblast, amniotic cavity. Then the layer of the epiblast, that is primitive ectoderm, and hypoblast, that is the primitive endoderm. Both these are in contact with structures epiblast, hypoblast, that is endoderm, ectoderm. Both these form the bilaminar embryonic disc, and this disc further forms the embryo, proper embryo structure. From this embryo disc, embryo develops, which is about 2 to 8 weeks. After 8 weeks, the embryo is known as fetus, and after birth, fetus is known as baby. Okay, see here, embryonic disc, formation of extraembryonic coelom, and this one is the cavity which is filled, blastocyst cavity is filled with the coelomic fluid, that is coelom. Okay, here is the uterine cavity, blastocyst cavity. All these about the structures of the gastrula. Again come to the point how development takes place. Zygote, first meridional cleavage, that is longitudinal cleavage, 30 hours after fertilization. Cleavage becomes complete and forms 2 blastomeres, 2 cell stage. Then 4 celled stage. Second cleavage is also longitudinal but right angles to the first one. Third cleavage is horizontal, 8 cell stage is formed. First cleavage forms 2 cell stage. Second cleavage forms 4 cell stage, and third horizontal cleavage forms 8 cell stage. Further 8 cell stage undergoes mitotic division. This division is rapid. Further subsequent divisions are rapid. Morula stage, 16 celled stage, which looks like a mulberry fruit. Morula stage. On 4th day, it reaches up to the isthmus region of the oviduct, then enters into the uterine cavity. Some inner mass of cells which are larger, while the outer mass of cells are smaller, clear cells. Inner mass of cells get attached on one pole, that is embryonic end, and other, that is known as abembryonic end. Early blastocyst stage converted into late blastocyst stage. In this stage, a cavity is created, that is known as blastocyst cavity, in which the uterine milk, glycogen-rich fluid get absorbed. Up to this is the blastulation process. This is all about the blastulation from zygote up to the late blastocyst stage, that is known as blastulation. After blastulation, formation of gastrule, that is gastrulation. late blastocyst stage. See here what happens. The free end of the inner mass of cells, here free end of the inner mass of cells forms the endoderm layer enclosing the yolk sac, then blastocyst cavity, and the remaining inner mass of cells form the ectoderm layer. Some ectoderm cells divide, redivide, and form the amniogenic cells. They enclose this cavity, amniotic cavity. This amniotic cavity contains amniotic fluid. I will tell about this amniotic cavity amniocentesis in amniocentesis technique. See here, this is the blastocyst cavity, endoderm layer. This one is the amniotic cavity. Ectoderm and endoderm are in contact with each other, embryonic disc. Chorionic villi are nothing but the syncytiotrophoblasts. With the help of chorionic villi, they get implanted into the endometrial wall of the uterus of the mother. Amniogenic cells, amniotic cavity, and ectoderm. All these structures form for the seer embryo. Embryonic disc further form the embryo. Here is the yolk sac. See here. Further, embryonic disc is further forming the three layers: outer ectoderm, middle mesoderm, inner endoderm. All these structures further forms the embryo. Yolk sac, amnion, amniotic fluid. This amniotic fluid is most essential in amniocentesis technique. Okay, if amniotic fluid is there is no amniotic fluid, there may be desiccation or maybe death of this embryo. So amniotic fluid is essential surrounding the embryo. Embryo further get converted to fetus.